And our next speaker is Aleksandra Walczak from the École Normale Supérieure de Paris. And Aleksandra is going to talk to us about taking or being precise while being in a rush. Is that so?
Yes.
Thank you.
Yeah, so I mean, basically, I know we're all starting to be tired and saturated. So if you need something, the one thing that I want you to retain is I'm going to add time to the problem. And basically, everything everybody has talked about, I'm just going to add time and worry about time and not having enough of it. And this came out with a long-term collaboration with Nathalie here, and then we were joined by Jonathan and Massimo thinking about it. You can't see my pointer. Yeah, here we are. And then Thierry and Tarek, and then it led back again to an experimental collaboration with Mirna and Mathieu. But I'm not going to tell you that story in that order. I'm going to start off with the fact that cells live in noisy environments. They have to avoid danger. They have to deal with temperature, food sources, and other stresses. And they build noisy representations of these environments. So given that there are these noisy input signals and cells have these representations, how can they read these noisy signals in a way that they still make reliable decisions? That's the question that I'm going to talk about. And do cells have enough information about the signal, and how do they make these representations to have enough information to make these reliable decisions? So I'm going to start with a simple system, which is a signaling pathway, which is the MAPK pathway and its specific subcomponent, the ERK pathway. And depending on whether you like development or whether you like the immune system or you like stress response, you've seen this beast and you probably hate it and your stomach churns because it's complicated. But at the end of the day, all that matters is that usually it functions as an on-off switch. That's what you're told. There's some signal, no signal, it responds, it doesn't. And the question we're going to ask is, how many distinct readout levels can it actually distinguish? Can it go beyond the switch? So back to what Thomas was saying, typically when you look at signaling cascades, you think about a one time point measurement, and you're just asking whether it can distinguish different outcomes. And in terms of Shannon information, this is 1 bit. So, but can you actually can this pathway actually distinguish more doses? Can it have different responses due to different doses? So here's an example with 4, which gives you 2 bits. And the question that we're going to answer is, where is the information? Where does it encode this information? And so it turns out, if you go beyond the typical view of just this one time shot, this measurement at one specific time point, and you look at traces, you can get more information in a trace than you get in a stamp shot. So this is an experiment, again, in ERK from some time ago, that if you look at a specific time point, you get less than 1 bit of information. But if you look at the whole temporal vector, it can go up and you get a little bit less than 2. And this was now later again studied in ERK in a slightly different context, actually a very interesting context that puts the cell in a context and says, well, we're not just in this artificial setting with one cell, but there's actually the environment. But if you just look without the environment, you still get a little bit more than a little bit less than 1 bit of information for the same signaling cascade. And again, we know this is a cascade that's involved in many different things. But all of these previous approaches have a lot of experimental limitations. They have a limited number of resolved inputs, small samples. They don't cover the full dynamic range, but that's not the issue I want to raise. The issue I want to raise is that there's also cell heterogeneity. All of these previous measurements were done at the population level. And we know different cells can behave differently, even different identical cells can behave differently. So in terms of this what we're looking here, they can have different, slightly different input-output relations or dose response cases. So what Mirna did is she decided to study this problem at the single cell level. And so the input to this cascade is FGF. Again, depending where you come from, you know and you love FGF. And what Mirna did is she replaced it by opto-FGF. So basically she used optogenetics to have a very precise control of the concentration of FGF that goes on. Okay, which means she can vary it precisely and she can turn it on and she can turn it off. And so that's what Mirna did. And then she measured the output of this MAP kinase cascade, which is proportional to ERG level. And she looked at the, basically, she looked at the nuclei and she saw how much of this reporter that is proportional to ERG is expressed. So what she did is she stimulated these cells with 7 doses of different amplitude, so basically increasing the light level, so which is proportional to increasing the FGF concentration. And she let the cells break in between the doses, and then she repeated this 20 times. So this experiment took a couple of days. Okay, this is painful. And then she got the output, which I said is this reporter of ERG. This is what the curves look like. But because these are the same cells stimulated in the same environment, she could basically get reproducible distribution. So we're back to what Thomas was talking about. And then we used machine learning to machine learning-based algorithms to estimate the mutual information from that. But the important thing is we're able to get the input and output distribution here, so we can perform this, and then it's sort of technical details. Okay. So what do we get? So this is the mutual information in bits between the dose, so the light dose, or if you want to think about it as FGF, and the output as a function of time. And we see that there's a peak around the 6th and 8th minute time points, which sets a time scale for the response. And the green line here is the information from individual single cells, and the blue line is from the population of cells or pooled cells. And we immediately see that individual cells have nearly double the amount of the information that pooled cells. So single cells are able to discriminate, to tell you what the input concentration is way better than a population of cells. That's what this result is telling you. And then we can group the data and look at time intervals. So we can look at what happens at 1 time point at 2 minutes, and then what happens between 2 and 4 minutes, 2 and 6 minutes, and so on and so on. So we're basically taking intervals of the traces and seeing where the information is. And again, population level in blue, single cell in green. And by comparing these high time points, which are still below 1 bit, to the individual cell data when you take intervals, you're able to see that there is more information in trajectories than in single time points. Basically, even the most informative single time point doesn't come close to the information you're getting from a trace. So by integrating over time, you will look, taking into account more time points, cells actually have more information. This is true for single cells and pooled cells. But the distinction that you always have more information in single cells remains, with the caveat that single cells are very heterogeneous. So you have some cells that really perform very close to the theoretical bound of being able, we have 7 input doses, so that gives us 2.8 bits, and some do worse. Okay, so there's huge cell-to-cell variability. So the information, there is much more information in a trace. If we ask where is the information is in a trace, than the information that is in a single time measurement. Of course, there's still information in single time measurements. So we can now turn the question around and say, well, okay, so now I'm measuring the output. Can I build a decoder. Basically, can I figure out by measuring the output what the input was? And I can build a universal decoder, meaning I take the outputs from all the cells and then I try to ask each cell, I give you this machine, this decoder, tell me what the output was. And if we do that, then we see that basically the universal decoder works better for some cells than for others. Again, huge heterogeneity, but generally doesn't do very well. So cells, it doesn't allow us to figure out what the input dose was. However, if we build an individual decoder for each cell, basically we train each machine for each cell separately, then it's perfect for all cells. So we have perfect decoding at the single cell level. So really information processing here happens at the single cell level. And so then we also asked, well, are cells that are closer to each other, are they more similar? Is there any information like that? Is there any correlation like that? And so we remember we saw this distribution and we don't see, this is colored by information, and we don't see really a correlation between where the cells are and how much information they have. And pushing that further, we asked, well, the daughter cells are more similar in their information processing capacities than randomized cells, and that's not the case. So basically, we're getting to the point where cells, in this setup, we see that single cells are able to process information better. Basically, they have their own ways of making a readout. This huge heterogeneity, it's something intrinsic to the cells. It's not linked to sort of some baseline noise in output levels, but it's not linked. It's not linked to some sort of lineage structure or spatial structure. So what we saw here is that cells have more information in a single trace. And so how is the fact that cells have a representation of a trace, because they express mRNA, the mRNA they express is some sort of representation, how does it allow them to make reliable decisions? So this brings me back to the fly, and as I said, the sort of starting point for all of this. The fact that when the Bicoid gradient controls the expression of hunchback, and this results in this precise boundary where cells at the boundary are able to result in different decisions, which means to read out hunchback or not read out hunchback based on a 10% difference in the Bicoid concentration. So the input concentration differs by 10%, which translates into 70 molecules, and this results down the road to different cell types. And this is done in a noisy environment. Bicoid has to find the binding site, diffuse to it. It's not a simple problem. And sort of 50 years ago now, Berger and Purcell put this together, and then Thomas and Bill put this together again specifically for the fly, and it sort of depends on the parameters, but you get the estimate that you need of the order of 40 minutes to do this with the precision that you explain in experiments. But the early cell cycles have about 5 minutes. So with Nathalie, we looked at this in detail, and she basically built concurrently with Thomas an MS2-MCP reporter, which allows you to track mRNA, and you see that this is a chimograph, so this is time and this is position. And even in the very early cell cycle 11, you get this precise boundary forming within 3 minutes of the start of the readout. So you get reproducible and well-defined decisions at the mRNA level, or meaning high and low hunchback readout in 3 minutes. So this led us to take a step back and sort of think about the problem differently. So what the Burke and Purcell scheme does is basically you give yourself a fixed amount of time and you measure binding and unbinding events, and you ask, you basically treat your gene as a measuring device, as a receptor, and you ask, based on that, based on how I'm collecting my data, do I think I'm in a high concentration regime or low concentration regime, right? Everybody agrees with that? I have this binary decision, and I'm trying to make it in a fixed amount of time. So instead we said, what if I don't have a fixed amount of time, but I'm measuring, and as a function of time, I'm comparing the likelihood that I'm in a high concentration regime or low concentration regime. So as a function of time, I update the likelihood between these two scenarios, and at some point, I reach a confidence level that I'm happy with. And then I say I make a decision. Okay? So this is my log likelihood, my two scenarios, I compare this, and this basically leads to a random walk in decision space, which is driven by a deterministic bias and some noise. And I stop it when I reach a prescribed error. So this is a mean first passage problem, which can be solved. And in fact, everything is basically that time to a decision is then inversely proportional to this bias, which, for those who care, is proportional to the Fisher information rate. So, okay, so Fisher information is another measure of information, which basically tells us how the parameter we're changing, in this case, the log of Lecuit concentration, is informative about the difference between the two scenarios. So very similar to what Madan was talking about. Okay, so the bottom line, time is inversely proportional to the bias. The stronger the bias, the faster the decision. So the blue one has a stronger bias. The decision distribution is moved to shorter times than the red one, which has a shorter bias. So stronger bias, better discrimination of concentration, faster decisions. And so then we can optimize the parameters for a fly system with 6 binding rates. And we can ask how fast can you make the decision? Specifically, can you make it in under 3 minutes? We have different rules for what constitute activation, how many binding sites you need to have bound for the gene to turn on. So, but basically for most of these cases, we see that we're able to make a decision reliably in under 3 minutes without any problems. We also asked if we can make it with a sharp boundary. This makes a decision harder, but it is still possible. So basically there's plenty of time to make this decision if we change the way we make a decision, going from fixed time to variable time. But remember, we still have the representation of the signal, which is basically the trace of the mRNA. And so that's the question we had is, well, can we implement this in a real system? Can we get so the log likelihoods have contributions to the off time and the on time that have certain forms that we need to fulfill. So basically, our molecular system will have to have inertia. And so there has to be some time delay. This is quite easy to implement in gene regulation. And it needs to forget. So mRNA needs to be degraded, which in this biological system maybe is a little bit harder to reproduce. But basically, there are schemes for making this happen. So at this point, we sort of went back to thinking about the trace, because it seems that this representation of a trace is something the cell has, but it doesn't mean this is what the cell has to use to make a decision, to make a readout. So far in what I just described, we assume that it does use the full trace, but it could be something different. So here we turned, and this is work with Tarek, who's here. We considered a slightly more complicated system where we have the transcription factor binding site, but we also explicitly look at mRNA binding. So we now, for one binding site, we have a 4-state system. And we compared the decision based on the full trace to a decision based on a reduced readout of the trace, which will be accumulated mRNA. Okay? So if I think of the trace as being my representation in the cell, then I say, well, maybe I will be making a decision to express or not express on some further statistic of this trace. So how does that change how we make the decision on basically what parts of information we use? And I think this is an idea that we heard this morning too. Okay, so now we're looking at this Fisher information, which is proportional to bias, as a function of the concentration we're trying to measure. So these are high concentrations and low concentrations. In red, this is the how fast we can make the decision based on the full transcriptional trace compared to, in black, the cumulative mRNA. The higher it goes, the stronger the bias, the faster the decisions. And so we see that in high concentrations, of course, decisions are easier, noise is smaller. Low concentrations, decisions are harder, so are harder, so slower. So, but if we look into so we can basically make faster decisions by exploiting the whole transcriptional trace. But if we look at which circuits are optimal for exploiting this information, for making this readout, they're very different in the two cases. So this is the optimal circuit for when you basically have the whole trace at your disposal. This is the optimal circuit when you only have the reduced statistics of the accumulated mRNA. And so when you have the whole trace, in both cases, the name of the game is basically to return to the off state. So the way you're not bound and you don't have the polymerase bound. Because you want to be unbound to be able to sense concentration, to bind the transcription factor. You want the transcription factor not to be bound to basically be able to function as a receptor. Otherwise, you're stuck, right? You can't sense anything. So in both cases, you want to return to this off state as fast as possible, because that's the only place you can you can make a measurement. And so basically in the transcriptional trace, you go through the network as fast as possible. And I should add another thing, which is actually very important, and I forgot to mention, that we do all of this optimization assuming constraints on rate. So we assume binding is diffusion limited, but we also assume other rates have maximum values. Otherwise, you could go through the roof. And this does change the results significantly, so you basically max out the rates. However, if what you're trying to read out is accumulated mRNA, well, the mRNA trace has, by default, information about the on state. It's encoded there. You can't get rid of it. So what you try to do is you try to basically reduce the variance as much as possible and use it as best as possible. And that gives you this back rate, because then by going back and forward here, you're actually able to use that. You modulate your on periods depending on the concentration of the ligand, and you get information from that. So you're forced to use them, so that's the difference. And this translates into different activation functions. So if you use the full transcriptional trace, you're not cooperative at all. You have your sharpness or steepness remains small. The Hill coefficient is less than 1. But if you're reading out accumulated mRNA, you generate effective cooperativity through using this 4-state system. But steeper, what this tells you, so you're way steeper with the accumulated mRNA, which doesn't mean you're more precise, because you're more precise with the full transcriptional trace. So in this fly field, there's also this idea that you want to be steep and precise, and it's the same thing, and it's not. All right, so let me then bring this back to the fly, and this is very much work in progress, and what we're trying to sort of figure out exactly what's going on, but these are measurements from this is data from Thomas's lab with the analysis of Ben Zoller, where they, for different gap genes, they looked at the time spent in the it's actually on-off states and the effective time spent in a cycle. And the interesting thing here was that this effective time spent in a cycle was constant. And so we looked at our two models, and because of this concentration dependency on state, it's the cumulative RNA model, which is here denoted in this dashed line, that reproduces this data and gives us this effectively constant readout, where this transcriptional trace always gives us a fixed on time. It doesn't give us these dependencies we see in data, and so it actually doesn't give us an effective constant rate. So long of the short, it's the cumulative mRNA that has similar signatures for the time spent in the cycle to what seems to be going on in gap genes. And so maybe a point to make here, just because the information is there and you could be making a faster, more precise decision, that doesn't mean that that has to be what the biological system does. We're not saying it's actually using cumulative mRNA, but it's at least more consistent. But the information in principle is there, and it could be used by other processes. And that's why I think it's important to distinguish between this representation and what is then read out. But if we go with this cumulative mRNA readout and we put in 6 binding sites and assume we want this cumulative decoding with a Hill coefficient of at least 5, we see that we can still make it with concentrations of where the bicoid system functions in under 3 minutes. So, okay. So to sort of summarize, what I tried to show you is that there is much more information in trajectories and snapshots. So maybe it's sometimes useful to think about trajectories. Then we have internal representations of input, the mRNA trace being a trajectory version of that, but we can then have different projections or readout, which give us both different information and different optimal molecular encoding. And what does that mean with different molecular encoding? It means different architectures. It means different promoters. It means different structure. And so single cell decoders are generally more accurate than universal decoders, at least in the setting that we show. And this can give us putting this all together, we can get real-time decisions that are fast, and real-time decisions can actually be much faster than fixed-time decisions. Okay, thank you.